So I have the pleasure of introducing our Grand Rounds speaker today. I've known Susan for more than a decade. I'm not sure how much more, but for quite a long time, 13 years approximately.
13
but who's counting?
years.
As many of you know, our department has a lot of research that goes into understanding how to minimize the impact of addiction for individuals and for society. And Susan has been working on understanding the basic biological mechanisms from a molecular as well as an experimental psychology mechanism for many years. Before she came to Seattle, she was at the University of Michigan and did her undergraduate work there as well as her PhD in the lab of Terry Robinson, who's a world famous expert in addiction. And then I was fortunate that she came to my lab for her postdoc. And that was because I didn't really know anything about how to do the experiments correctly. And she would very gently remind me why we're doing the control experiments and how we had to do things properly. And so she had a very positive effect on the work in my lab. And through the time that she was in our group, she was getting some great grant support and great papers, and then went on to start her own lab at Seattle Children's Research Institute and attracted graduate students and postdocs, and has been branching off into directions that she'll be talking with us today about. But I think that we're very fortunate to have her in the department, and she is recently, we believe, being promoted to associate professor as long as she keeps out of trouble for the next couple of months. So join me in welcoming Susan Ferguson.
Great. Can you guys hear me okay? Okay, well, thanks, John, for that introduction. And it's a real pleasure to be back here. As John said, I did my postdoc here, and so I have very fond memories of the research and training building. And so today, as my slide here shows, I'm going to be talking to you about how we're using animal models to study and map the neural circuits that are involved in different aspects of addiction. And so I wanted to put up if I can get this to advance forward. Okay, it was working a minute ago. So do you know how to get it at all to advance?
The clicker.
The clicker. Okay, so just.
Oh. Sorry. So.
Okay. Oh, this seems to be working. Did it just?
Okay.
Okay. Sorry about that. So I wanted to put up the CME learning objectives. So given this is a grand rounds and many of you are clinicians, I am going to be spending a fair bit of time throughout my talk. I'm just sort of describing different animal models that we use to model addiction. So how do we do that? How do we capture the different aspects of addictive behavior in our animals? And as well, I'm going to talk to you about some of the ways, the cellular and molecular tools that we have to both visualize activity in specific cells in the brain, as well as to manipulate the function of those cells to try to probe their role in aspects of addiction. And then finally, I'll be telling you about, of course, the work that I'm doing in my lab, which is to better understand the role of the striatum, cell populations within the striatum, as well as different inputs from the cortex and thalamus, and how those regulate addiction behaviors. And certainly feel free to interrupt me if you have a specific question throughout the talk, or we can discuss it at the end. And so as many of you here, I'm sure very much so appreciate, addiction is really this chronic debilitating disease that's has sort of these hallmarks that we see in addicts across different drug classes. And those include an escalation of drug intake. So addicts, of course, will take more and more drug over time, often more than they intend to. They'll also use drugs despite negative consequences. And these can be consequences to themselves or to others. And we also see with addiction that a very sort of compulsive drug taking and drug seeking behavior will develop over time. And as a result, it's really hard for addicts to stop, even if they want to. And then finally, one of the biggest sort of problems that we have with respect to treatment and successful treatments is that addicts have a very high chance of relapse. So the majority of addicts will relapse at least once, if not multiple times, through the progression of their disease. And so what I'm going to be focusing on mostly today is talking about cocaine addiction. And cocaine use is still very much prevalent in our society today. So we have an estimated 1.5 million current cocaine users in the US. And when we look at the number of emergency room visits, the 1.3 million visits that we see for drug misuse or abuse that show up, in the emergency room, 40% of those involve cocaine. And approximately 5,000 people overdose annually as a result of cocaine. And then I wanted to point out that of those 1.5 million users, approximately 913,000 of them meet the DSM criteria for cocaine dependence or abuse. And so I think that's a really important point, and that is that not everybody that's using cocaine is really classified, according to our criteria as an abuser or an addict of drugs. And so if this set of emojis sort of represent those individuals that have tried a drug, only a subset of those will go on to participate in sort of casual or recreational drug use. And then this set of emojis represents the number of individuals that have initially tried drugs that'll go on to become abusers. And finally, this set, which is about 15 to 20% for cocaine, will go on to meet those criteria for addiction. And so one of the things that we're really trying to focus on in the lab is what are the neural circuits that sort of mark this transition to addiction? What is it about some of these people that can sort of participate in recreational drug use, but are resilient to developing addiction? And, on the flip side, what happens to the neural circuits of these individuals that after they use drugs will sort of propel them into the cycle of addiction? And so, as I said, I'm a basic science researcher. So the way that we do that are to develop and use animal models to study addiction. And so there's sort of broadly two classes, I guess, if you will, of models that we use to study addiction. One set of those are sort of experimenter based with respect to administration of the drug. So the experimenter gives the animal the drug, has sort of control over the drugs, and this is in contrast to other models which are called self-administration or self-administered models, where the animals have control over their drug use. But we have a lot of value still in these experimenter-administered models. Two of the main ones that people use today, one is called conditioned place preference. And so with conditioned place preference, there's basically, this is used to measure the rewarding effects of drugs. And so you've got this apparatus that has 2 or 3 chambers. And what happens is you give an animal an injection of a drug like cocaine, and then you put them in one side of this box. And then in other sessions, you give them injections of a neutral stimulus like saline. You confine them to the other side of the box. And then on the place preference test, the animals are allowed to freely move among these chambers. And the idea is if they found the drug to be rewarding, they'll spend more time in that place that they've associated with that positive experience. If they find the drug to be aversive, they'll avoid that side and they'll spend time in the sort of neutral paired side. And so we can use that, as I said, as sort of this index of reward. We can use this to probe whether new compounds are rewarding to animals. We can look at the different circuits in the brain and try to identify new components of the brain circuits that might be involved in reward. Another one of these experimenter-based models is called psychomotor sensitization, and I'll be showing you some data that we've been doing in the lab to use this model. But basically, this model refers to giving animals injections of the same dose of a drug, such as amphetamine, and you can see that the behavioral response to this same dose increases over time. And this is in contrast to something like saline, which shows a very low locomotor response on the part of these animals that doesn't really change. Another way we can look at this phenomenon of sensitization is to withdraw animals from the drug and then give everybody what's called a drug challenge. And you can see that animals that have gotten a history of drug treatment, so these are animals that were pre-treated with amphetamine, they show this greater behavioral response to now this challenge injection of drug compared to these animals that are now seeing that drug for the first time. And with respect to using this as a model, it's certainly the case that people do show behavioral sensitization. So for example, amphetamine addicts will show a sensitized eye blink response that they develop over time. But really, we're not using this with respect to a true model of the human condition. What we're really interested in is this is sort of an easy behavioral readout for a sensitized neural circuit. So the neural circuits that regulate this behavior in animals are the same neural circuits that become compromised in addicts. And so again, we can kind of use it as a basic model to identify specific components of the circuitry of addiction and probe whether we can change this kind of behavioral readout by altering that circuitry. Okay, so in contrast to these experimenter-based models, we also use self-administration-based models in the lab. And so these are all models where the animal now has control over their drug intake. So what happens is they are put into this chamber called an operant box, and these animals can press levers or do nose poke responses in order to get IV infusions of a drug like cocaine. So they put an IV catheter in the animals. And so in that way, the animals really have control over their drug use, just like a human addict has control over their drug use in the sense that they're the ones giving themselves the drug. And then we can do a number of different kinds of manipulations to modulate the paradigms that we use with this model to then produce addiction-like behavior or not. And so one of the ways that we do that is to alter the amount of access that the animals have in any given session. So sort of a classic self-administration paradigm takes place over approximately 2 hours, where the animals can go in this box and they can lever press to get these infusions during a 2-hour period. However, more recently, other researchers have been using what's called long access, where instead of 2 hours, the animals go in for a 6-hour period. So it's thought to sort of mimic binge-like drug use in people, and we see sort of these addiction-like behaviors developing, like escalation of drug intake when we put the animals in for longer periods of time. Another thing that we can modulate in these animals is the duration of total sessions that the animals have in these boxes. And so again, these sort of classic experiments that people run have a fairly limited duration. The animals will self-administer for 7 days or for 2 weeks, and then we'll look at the circuitry. Well, more recent models have used what's called an extended duration, where the animals self-administer these drugs for much longer. They'll do it for a month, 2 months, 3 months. And what we see is when animals are given this opportunity to have a much longer cocaine experience, we can start to see in a subset of animals, and I'll show you that soon, develop these sort of addiction-like criteria. And then finally, another thing that we can sort of modulate in these paradigms is in any given session, the type of access that the animals have to the drug. And so this is called continuous versus intermittent access, and it's shown here. So this is that operant chamber I was telling you about, where the rat, in this case, can press these levers to get these IV infusions of drug. And in sort of, again, these sort of classic experiments I'm referring to, the majority of self-administration paradigms are animals undergo what's called continuous access. So with continuous access, these are showing brain cocaine concentrations of the rodents. And what happens is there's this initial loading phase where the animal goes into the box, they'll lever press a bunch to get to a certain level of brain cocaine concentration. And then throughout the session, they are really good at sort of titrating their response to maintain a very steady state level of cocaine throughout the session. And as it turns out, this isn't really the way that addicts take drugs. And there might be a number of reasons. Addicts might have a limited supply of drug, and so they kind of figure, well, maybe that's not the best way to sort of feel high all the time. And so what we can do is more mimic the way an addict will take a drug by this intermittent access paradigm. So with this paradigm, animals go into the box, they have 5 minutes where they can take as much drug as they want, and then they have these forced periods, what we call of signaled unavailability, where a cue comes on to tell them, for 25 minutes, you can't get any drug. And then after that period's over, there's another 5 minutes, they can get drug, then they've got another period of off. And what that does to brain cocaine concentration is cause these spikes in cocaine concentration. And so you can imagine, just even at the cellular level, having continuous cellular access, if you will, to a drug versus these sort of off and on cycles are going to have very different neurobiological consequences. And so what we've been doing in my lab is to pair this intermittent access paradigm with that very extended duration to sort of look at how this changes the behavioral consequences, if you will, of drug self-administration in our animals. And so what we do is animals undergo 40 sessions of this intermittent access, and then we classify them along 3 criteria of behaviors that sort of have evolved out of DSM criteria. So one thing we look at is how much drug seeking the animals will do during these periods of signaled unavailability. So during time where they can't take drugs, how many times are they lever pressing to try to get that drug? We also look at how motivated the animals are to take drugs. So addicts show very high levels of motivation to get drugs. The way that we do this in our rodents is to do something called a progressive ratio schedule of reinforcement. So for this schedule, the first time the animal presses, he might get a drug infusion. But the next drug infusion, he might have to press 5 times. And then the third drug infusion, he might press 15 times. And we keep increasing that work requirement for him in order to get a single infusion of drugs. And of course, the more motivated they are, the more they're willing to work to get that drug infusion. But at some point, an animal will say, you know what, this is just not worth it. I don't want to press 1,000 times to get one infusion of drug. And so every animal has a threshold. And at that point, at which they say, forget it, this is just not worth it. That's what we call the break point, the point at which they're no longer motivated to get the drug. And so we can kind of compare the motivation across animals using this metric. And then the last thing we look at is punishment-resistant drug use. So how willing are our animals to undergo a negative consequence in order to get a drug infusion? And so what we do is we pair our drug infusions with foot shock. So for this test, the animal might press a few times thinking he'll get a drug infusion. He gets a mild foot shock. Then the next time he presses a few times, he'll get a foot shock paired with a cocaine infusion. And so that cycle will sort of continue. And what we find is that most of our animals, who sort of participate in, let's say, casual drug use, say, you know what, this mild foot shock isn't worth my drug use, and they very quickly stop responding. But a subset of animals will continue to lever press to get these cocaine infusions, despite the fact that they're also having foot shocks. And so what we do, like I said, is we run the animals through lots of self-administration, and then we probe them for these 3 criteria and look at how high of a level of response do they show on these 3 metric. If they have a very high response, then we give them sort of a plus in the criteria for addiction, if you will, and we can sort of grade whether they have a very high response for all 3 of these criteria, for 2 of them, for 1 of them, or none of them. And so what you can see here is that we are able to sort of classify our animals into either a low risk phenotype or a high risk phenotype based on their level of response on these different behaviors. And these high risk phenotype animals show much higher behavioral responses for drug seeking, motivation, and for punishment resistant drug use. And these behavioral differences among animals show up despite the fact that these 2 groups of animals take the same amount of drug. So it's not just that these high-risk phenotype animals are taking lots more drug. They're not. They're taking the same amount of drug. And so whatever behavioral differences that we're seeing here are due to some sort of difference in the underlying circuitry. It could be that the low-risk phenotype are somehow resilient to these changes that drugs sort of do to the brain. It could be that in the high-risk phenotypes, these circuits are really being compromised by the drugs in some way. And so this enables us to be able to study the circuits of what's different about these 2 groups of animals. And the circuit in particular that we're interested in is called the cortical basal ganglia thalamic circuit. And sort of at the center point, if you will, at least in my world of the circuit, is something called the striatum. So the striatum, of course, acts as sort of an integration center. I'm showing you some different glutamatergic inputs here, as well as the dopamine inputs, but the striatum basically takes those inputs and lots of different ones. Serotonin, of course, is John's favorite, but it takes all of these different inputs and it synthesizes them to guide the behavioral output of the animals. But the way that it does so is through 2 primary output pathways. So in one case, of these cells in the striatum are medium spiny projection neurons, and one set of these projection neurons go directly to the output structures of this circuit. So based on that connectivity, very creative scientists call this the direct pathway. And we know from sort of classical studies of basal ganglia function with respect to Parkinson's disease, that activation of this pathway can act as sort of a go signal to facilitate behaviors to go forward. In contrast, we have this second set of medium spiny neurons in the striatum that indirectly project to the output structures through the globus pallus external and subthalamic nucleus. So again, based on that indirect connectivity, this pathway has been termed the indirect pathway. And again, we know from those classical studies of basal ganglia function that if we activate this pathway, it can act both functionally and mechanistically to oppose the direct pathway output to then act as sort of a brake or a limit on behavioral actions. And so, as I said, we're really interested in my lab in being able to study these different cell populations and these pathways and try to get a sense of their role in addictive behaviors. And one of the ways that we're doing that is using a technique called dread receptors. So you may have heard in other talks, particularly from John, about these receptors, but basically they're a class of artificial mutated receptors. The ones that we're using are mutated muscarinic receptors. So what that means is that they no longer have the same function as a muscarinic receptor in the brain, so we can express them through different methods. The main thing that we use are a viral-mediated approach. So with a virus, we can deliver these receptors into parts of the brain. They get expressed on different pathways or in different cells, but because they're not activated by any endogenous ligand like acetylcholine or anything else, when they're being expressed, they just sit there and they don't do anything. So they have very little to no basal tone. But when we give a synthetic ligand, a common one right now is something called clozapine N-oxide, a metabolite of clozapine. And this CNO has very high affinity for dread receptors, but in these rodent brains is otherwise inert. So it's not activating anything else in the brain. And so what we can do is use this sort of pharmacological system that we have to be able to transiently alter the activity of the cell population that we're targeting to see if what happens in these behaviors, if we turn down activity of a cell population or turn up activity or change parts of these pathways, how does that change the behavior of the animal? And the main dread that I'm going to show you and talk about today is the HM40I, or a GIO coupled dread. So this is our inhibitory dread. We know from a number of different studies that activation of this dread will inhibit or decrease neuronal firing. This is showing the intracellular signaling of that GIO coupled receptor. So we know that it can decrease cyclic AMP activity. When GERK potassium channels are present, this dread receptor will activate those channels to hyperpolarize the neurons. We've shown that it can decrease neurotransmitter release. But I do want to point out that when you're modulating signaling, GIO signaling, it's sort of a complex activity. It's not the same as another technique, optogenetics, which acts at channels to just block receptor or block neuronal activity. So in addition to this sort of cyclic AMP cycle, the dread receptor will also, on a slower onset, will activate a beta-arrestin signaling cascade to actually increase MAP kinase and ERK signaling. So what we're doing is modulating the cell activity, which mostly leads to decreases in neuronal firing. Okay, so this is actually work that I did when I was a postdoc in John's lab. So we wanted to confirm that using this dread, expressing it in our targeted cell populations in the striatum, that we could indeed detect decreases in neuronal activity in the cells that we were targeting. And we confirmed this with two different methods. One of those was to use the immediate early gene CFOS as a marker of neuronal activity. And what we found is that when we expressed and activated this dread receptor in those cell populations in the striatum, we could detect decreases in stimulated neuronal activity with that CFOS marker. We also, in collaboration with some other scientists, showed that when we could express and activate these dreads in slices, that the CNO resulted in this decrease in evoked action potential. So indeed, in the striatum, we were seeing this decrease in neuronal activity in the cells that were expressing this dread receptor. And so our very first experiments that we wanted to do was, well, what happens if we express these cells in that direct pathway or that indirect pathway? Could we modulate the development of amphetamine sensitization? That model I told you about, where you give repeated injections of the drug and the behavior should go up. Can we change that behavioral readout? And so the way that we did this experiment was to express those dread receptors either in the indirect pathway cells or the direct pathway cells with our viral vectors, and then we would activate those dreads right before we gave them the injections of amphetamine during this induction phase of sensitization. We then withdraw the animals, and on this sensitization challenge, we look at how we affected that behavior. And so this first experiment was what happened when we decreased activity of the indirect pathway. So we're basically taking off the brake on the circuit. So we would predict that we would see an increase in the behavioral response if we're taking that brake away. And indeed, that's what we saw. So these two groups here got the amphetamine injections. So we should be seeing the development of sensitization. In 4 treatment sessions, we're starting to see sensitization in our controls. And you can see we see this very robust sensitization to that same regimen when we take the brake away on the circuit. And when we withdraw the animals and do the amphetamine challenge, you can see that to those 4 injections, we're not seeing a whole lot of sensitization in our controls. but we're still seeing this very robust sensitization in these dread animals. So basically taking away the brake is facilitating the development of this sensitization. More recently in my lab, a graduate student, Kenichi, he said, well, what happens when we look sort of downstream of this indirect pathway at the STN? So the subthalamic nucleus, it's the neurons in that nucleus, they're the only excitatory node in this indirect pathway circuit. So what he wanted to see is what happens when we activate the STN. This should lead to an increase in brake activity. So we would predict you should see a decrease in sensitization if you increase the brake. And that is what he saw, which is shown here. So again, here's the animals that are getting the amphetamine. He's now doing a much longer treatment regimen, and you can see that the vehicle controls show this very nice development of sensitization that's blocked when he increases brake activity. And then when he does the challenge, you can see sensitization is very nicely maintained in our vehicle controls, but it's still gone. So we've been able to block this behavioral readout and presumably blocked the sensitized circuit by activating the brake pathway. So in addition to that brake pathway, we're also thinking, well, if these pathways sort of work in concert, we could also modulate the activity of the direct pathway to produce a similar kind of effect. So whether you increase the brake or decrease the go signal, the drive of the circuit, you should be able to block the sensitization. We were a bit surprised to find that's not exactly what we saw. So here now we're decreasing direct pathway activity, and even though we're doing that during this induction phase, we're still seeing sensitization develop similarly in our dread animals compared to the controls. However, and very interestingly, when we withdraw the animals and do this drug challenge, we still see sensitization in our controls, but the dread animals are now no longer sensitized. So the effect on the direct pathway isn't exactly in the opposite direction of the indirect pathway. It's having a more complex effect to sort of modulate long-term function. And so more recently, we've been really interested in trying to understand the role of the direct pathway using our addiction models. So this is work done by a former postdoc in the lab, Lindsay Yeager. And so what she did was to do this operant self-administration using that 3 criteria model that I told you about earlier. And she's now expressing the dread receptors using a slightly different approach. So she needed to have very long-lasting expression of the dreads. So what she did is put a dread receptor into the striatum that is Cre recombinase dependent. So it's in a flipped orientation, but in the presence of this enzyme Cre recombinase, the orientation gets flipped into the correct orientation, and then you get expression of this dread receptor. And so what she did is put a second virus, it's a retrograde Cre virus. She put it just in the downstream part of the direct pathway. And so now that virus goes back into the striatum, and she can get expression of these dread receptors only in the direct pathway cells. And so they're not in the indirect pathway cells. And of course, because this is a different technique than we had done in the work with John's lab, we wanted to confirm that we could, with this method, see decreases in the activity of these cells in the direct pathway. And so the approach that we use to confirm that is something called calcium imaging using a genetically encoded GCAMP6. And so this is basically a protein that in the presence of calcium induces a conformational change which causes fluorescence. And we can measure that fluorescence and get a sense of the activity of the cell. We know there's a very strong correlation between these calcium transients and action potentials. So if you see an increase in calcium, you sort of infer that that's an increase in cell firing or cell activity. If you see a decrease, then you'd be decreasing activity. And so again, just like we've done with those other experiments I've showed you, we're using a viral method to be able to express this GCAMP into cells. So this was work done by a current graduate student in my lab, Aaron Garcia. This is just a picture showing you that we're getting this GCAMP fluorescence signal in these direct pathway cells in the striatum. And so here's sort of the baseline activity of these cells. These are their calcium transients. And you can see here that when we put CNO, these are in slices, when you put CNO in the bath, you get this complete reduction of this calcium activity. And then when you start to wash that out, you start to see this return in activity. So we're confirming with this method that indeed we can detect decreases and shut down those cells when we activate this dread receptor. And so we then took this approach and we used that 3 criteria model to look at what happens in animals that we've classified as high risk and low risk when we turned around direct pathway activity. Our hypothesis was that we would sort of block all these addictive behaviors, and hopefully what you're appreciating here is we saw no effect. And so either at our low risk or high risk phenotype, across the drug seeking, the motivation, and the punishment resistance, we weren't changing the behavior of these animals. And we also weren't affecting their drug taking. So they were still taking just the same amount of drug as they had, as the green animals or the dread animals, as our control animals that aren't having this decrease in direct pathway activity. So the next thing we did with these animals is to look at relapse behavior. And so the way that we do that in animals is we take them through extinction. So you can kind of think of them as this as rehab. So animals go back in the box, they can lever press, but nothing happens when they lever press. And so over time, these animals will stop lever pressing for drug. They learn, well, there's no point in doing this because I'm not getting any drug. And just like with addicts, we can cause relapse, we call that in our animals reinstatement, by giving them exposure to a few different things. So if you give them exposure to a stress, just like with people, that can cause an increase in drug taking and seeking behaviors. If you give them what's called a drug prime, so a little taste of drug, that'll cause this reinstatement behavior. And additionally, during our self-administration, we pair those drug infusions with a cue. And so if you expose them to the cue that had been associated with the drug, they'll also start searching for drug again. And so what we did is take animals through this extinction behavior and then look at reinstatement to cues that were previously paired with the drug. And one thing to note here is here's our low risk condition, here's our high risk phenotype, and this is now, we're not doing any dread manipulation here, and you can appreciate that the high risk phenotype have much greater levels of this reinstatement or relapse behavior. So again, this is a confirmation to us that this model is sort of capturing pathological drug seeking, if you will, compared to what we think here is sort of normal drug seeking associations. And what we found is that when we decreased activity of that direct pathway, of that go signal, we were able to actually reduce this pathological drug seeking in this high risk phenotype down to what we sort of are considering normal drug seeking conditions. So we did an experiment in a separate group of animals to look at sucrose seeking and sucrose reinstatement. So sucrose is a natural reward for these animals, it's sugar, and you can do the same sort of thing where they learn to lever press to get sugar pellets. You can take them through extinction of that response, and then if you give them cues associated with that sugar, they'll show reinstatement of sucrose seeking. And interestingly, there was no effect on this reinstatement to sucrose seeking when we turned down that direct pathway. And the reason why an experiment like this is important, as well as looking at sort of normal drug seeking, is that, you know, for thinking of using these different cell populations and things as targets for drug treatment, as again, most of you being clinicians, I'm sure appreciate, is that people that just have no motivation for anything aren't likely to stay on some sort of treatment. It's not going to make them feel very good. And so if we could have a treatment that is selectively modulating this pathological behavior, but still maintaining normal reward value for things like sucrose, then that's probably going to be a treatment they're more likely to stick with. And so we were definitely intrigued by these kinds of findings. We're also curious in the lab to know whether these kinds of behavioral modifications of the circuit extend to other drug classes. So we've also been looking at the role of the direct pathway with heroin consumption. And so this is work that's been started in the lab by a graduate student, Tim O'Neill. And so what he did here is he used that intermittent access paradigm that I've told you about, and he's now giving the animal 6 hours of heroin access each day. And so he does that. You can see here, we see this very nice escalation of drug intake over these 13 sessions. And then what he's doing is delivering, just like Lindsay did, these dread receptors into this direct pathway with this combinatorial approach. And then on one test, he's doing these progressive ratio tests, so this motivation test to see what happens when we decrease activity of this direct pathway during the motivation to take heroin. Does it decrease that motivation? And then he's also looking at the reinstatement. So with respect to motivation, very similar to what we saw with our cocaine behavior, there was no effect on motivation. So the animals, even though their direct pathway was decreased, they still were as equally motivated to take drugs as our controls. And so then he took the animals through extinction, which you can see here, and then he looked at that cue-induced reinstatement. And very similar to what we saw with cocaine, here's the reinstatement with vehicle, and then when he decreases the direct pathway activity in these animals, we see this reduction in cue seeking. So kind of taken together, these experiments are consistent with our hypothesis that there needs to be a balance of activity between this direct and indirect pathway to get normal functioning and behavior to occur, and that drug use is leading to some sort of imbalance in activity of these two circuits that in a certain group of individuals leads to addictive behaviors. But what these kinds of experiments don't tell us is the nature of this imbalance. So is it an increase in activity of the direct pathway, a decrease in activity of the indirect pathway, or some sort of combination of both that's driving addiction behavior in addicts? And so that's a question that's of ongoing interest to us in the lab, that we're very actively studying it. And so I have some pilot data to show you with respect to what we're finding. And so this is again work done by Aaron, and he's using now the continuous access versus the intermittent access schedule that I was telling you about before. So some of his animals just take drug for 2 hours in the box. Some get this more addict-like pattern of drug intake. And as you might appreciate, in this case, these animals that have a full 2 hours of box time with cocaine versus this is about 25 minutes, they take a lot more drug. And so it's a little bit of a confound in our experiments. However, what's interesting is that despite the fact that they take almost twice as much drug, it's this group of animals that actually show more of that pattern of addiction behavior when we start probing them on some different behaviors. So for example, our continuous access animals, they don't really escalate their drug intake. They sort of keep this stable level. Whereas that escalation, which is a hallmark of drug use, we see this rapid escalation in our intermittent access animals. In addition, if we look at that locomotor sensitization, which is, again, the idea that the circuit behind this sensitization is sensitized and enhanced. In our continuous access animals, we don't see sensitization, but we see this very robust sensitization developing in our intermittent access animals. And so what we've been doing is to go back to that calcium imaging that I told you about earlier and to say, well, what happens to the activity of these cells in these two groups of animals? So we take these animals, they undergo the self-administration, and then we can start to look at striatal cell activity. Does it increase at baseline? Does it change with exposure to cocaine? And so for these initial experiments, Aaron just put the calcium indicator into all cells in the striatum, so both those indirect and direct pathway cells. But what he noticed is that if we look at just in control animals that have never undergone cocaine, we observe two types of cells with respect to how much activity they're showing. So we see these sort of this class of cells that show relatively low cell activity, and then we see these other cell populations that show about twice as much activity. When we go to the literature, we know that these direct pathway cells have much lower baseline levels of activity than the indirect pathway cells. So our hypothesis right now is that we're marking sort of direct and indirect pathway cells with his studies. Right now, he's sort of going back and actually putting the GCAMP into these selective cells to see if this hypothesis holds. But here's his preliminary data that he has collected so far. The purple bars here are animals that have no cocaine experience. These white bars are the continuous access, and then these are those intermittent access, more addict-like animals. And so these are the low activity cells, the high activity cells, the sort of direct pathway cells, indirect pathway cells. And the first thing to note here is at baseline, the activity of these cells haven't really changed. So these, regardless of what kind of experience they've had in the past, the activity looks pretty normal. But then we looked at what happens when you put cocaine into the bath. What does the activity do relative to baseline following this cocaine? And what you can see here is that in the intermittent access condition, in these sort of potentially direct pathway GO cells, we see this huge increase in the percent change of activity when cocaine's put into the bath. In some cases, I mean, it went up to 600% increase in activity. But that was only in this intermittent access condition. At the same time, if you look at these high event cells, so these are sort of the indirect pathway break cells, sort of across the board, even in control animals that have never seen cocaine, we sort of just see a reduction in this indirect pathway activity. And so, as I said, these are experiments now that Aaron's going back and doing much larger ends and actually being able to mark these two cell populations. But it's leading us to the hypothesis that with drug exposure, just sort of across the board, regardless of whether you're an addict or not, you're going to reduce your indirect pathway activity. And so this kind of fits with the fact that we saw a really strong modulation of those behaviors when we started modulating indirect pathway activity. However, we think there's also this concomitant increase in direct pathway drive that's sort of selective for our animals that develop addiction. And so it's this combination of factors that is probably driving addiction in these addict-like animals. Okay, so in the last bit of my talk here, I'm gonna switch gears a little bit and tell you about some of the work that we've been doing with some of the glutamatergic inputs into the striatum. And so a lot of the striatal activity is gated by these excitatory inputs for different brain regions. And one region that has been very strongly studied in addiction is the prefrontal cortex, which sort of acts as an executive control on the circuit. And so we know a lot about what the prefrontal cortex as a whole does, but we don't know that much about what cortical inputs specifically into the striatum are doing in addiction. And so another graduate student in my lab, Amanda Wunsch, she had studied this as part of her dissertation work. And so she again used that combination viral approach that I've showed you now a number of times to be able to express these dread receptors only in those cortical cells that are going into the striatum. She's now targeting a part of the striatum called the nucleus accumbens. And one thing that we noted is these cortical cells that go into the accumbens, they can send projections, collaterals, into other parts of the brain, and other parts of the brain that they can potentially send projections to also go into the striatum. So the first thing we wanted to do is kind of check with our approach, well, how specific were we for targeting the nucleus accumbens? And you can see here that we get very strong expression of these dread cells in the nucleus accumbens. We did also see some collaterals in the amygdala, which is another glutamatergic input into the striatum. So we next confirmed that our manipulation was still selectively affecting these cortical cells into the accumbens. So we looked at stimulated CBOS. And what we found here was that when we activate these dread receptors that are supposed to be projecting from the cortex into the nucleus accumbens, we could in fact detect decreases in activity of cells in the nucleus accumbens. So our manipulation was decreasing activity in the accumbens, but when we checked in the amygdala, there was no effect. And so we're pretty confident with these experiments and others that we're selectively modulating cortical input into the accumbens while sort of keeping cortical input to other structures unaffected. And so with these experiments, what we did was that viral approach that you've seen now. The animals underwent self-administration, and we looked at the progressive ratio test again during the self-administration. We took the animals through extinction, and now this time we're using a drug prime to reinstate the drug-seeking behavior. So we give the animals a systemic injection of the drug. It sort of brings up interoceptive and extraceptive cues that are associated with that drug use, and they start looking for more drug. And so this is just their behavioral response in the 3 sessions before we did this manipulation. So here's both the controls and the dread animals showing normal motivation to take drugs. And then what we found is when we decreased activity of these cortical input into the accumbens, there was no effect. So again, motivation seems to be a very hard thing to modulate in our animals. But we went on with these animals and we took them through that extinction behavior, which is you give them the lever pressing, and over time they learn that that doesn't do anything. And so this is just our GFP controls here. So you can see on the first day and to some degree the second day, they're still lever pressing, but over time they very quickly stop. Well, one thing that we found that was quite unexpected is that in our dread animals, so we're not doing any manipulation at this point. They had that manipulation during the motivation. You can see that their extinction is much different than the controls. So they're much more reactive. They have much more higher levels of drug seeking in these first few days of extinction. They seem to be, and there was something about, changing about how they're associating those cues with the drug to drive their behavior that's different than the controls. So this was very intriguing for us, but we did observe that over time, they show normal extinction curves by the end of the 10 sessions. So we took them on to that reinstatement phase. And so again, this is a regular control animal, and there's their reinstatement, their drug seeking following the systemic injection of cocaine. And again, to note, much like what we saw here in our dread animals, so there's no manipulation yet here, but this is just their reinstatement to a taste of drug. They're having a much greater response. So they're seeking much more. But during this phase of the experiment, if we decrease cortical activity, we can actually reduce that drug seeking. And so what is going on with this? Well, what we think this is sort of representing is that cortical activity is really important for making these drug-associated cues. And we know with humans, with addicts, that when they take cocaine over time, we see this hypofrontality develop. So at sort of baseline levels, their cortex activity is dampened down. And so we think that decreasing that activity during drug use is really critical by a number of different potential mechanisms, but it's critical for the formation of the association of cues, of associations between drugs and cues. And so what we think is by sort of doing that hypofrontality, if you will, during the drug taking, we sort of increase the associations that the animals were making between the cues and the drugs, which was being reflected in that enhanced extinction and reinstatement responding. But then we also know with addicts that when you expose them to cues, you see this really strong cortical reactivity. And so the idea with our reinstatement behavior is that when they were being exposed to a cue, they had this huge burst of cortical activity. When we dampen that down, it was then sufficient to decrease the responses by cues. So that's sort of driving relapse behavior. Okay, so in just the last few minutes here, I'm going to tell you about some very recent work that we've been doing in the thalamus. And the reason that we got interested in the thalamus is that there's been a lot of focus on the cortex, which is this glutamatergic drive into the striatum. But as it turns out, the thalamus also sends a very strong glutamatergic input into the striatum, and very little is known about that thalamic input and what its role is in this reinstatement behavior. And so the first thing, because we know so very little about the thalamus, what we just wanted to do was target the dreads to the thalamic nuclei just generally. So not modulating those inputs at this point, just modulating all output from the thalamus. And so we did this by putting those dreads into the thalamus. And you can see here with this fluorescent expression that these neurons are going to the accumbens, but also to the amygdala, the prefrontal cortex, and the hippocampus. So we're really sort of dampening down a widespread glutamate input from the thalamus to lots of different brain regions for these first experiments. So again, Amanda used the intermittent access paradigm, and you can see that over 14 sessions, she gets this nice escalation of drug intake with these animals. She took them through extinction, and then now she's looking at what happens if you decrease that thalamus activity to drug prime-induced reinstatement. And you can see she can decrease drug prime reinstatement as well as reinstatement here to a cue. So decreasing the thalamus in general decreases reinstatement to two different stimuli. But importantly, as I told you before, with the sucrose cue, she has no effect on the reinstatement. So it's not just changing overall motivation to all rewards. And because we're looking at decreases here, she confirmed that her manipulation wasn't just affecting locomotor activity. So you can imagine if the animals are just sitting in the corner not doing anything, that would be reflected here as a decrease, but the animals move around just fine. So then the next question she had, well, could she recapitulate those effects that she saw with the general motivation if she now decreased activity of thalamus specifically into that nucleus accumbens region? And so that was the last experiment that she did here. She did that same Cree recombinase dependent approach to get those dreads selectively in those thalamic cells into the nucleus accumbens. But one thing that we noted with her expression patterns is that anterior regions of these nuclei go to the shell region of the nucleus accumbens, whereas these more posterior parts of these nuclei go to the nucleus accumbens core. And there's a strong animal literature to show that the shell versus core regions of these of this nucleus accumbens area can differentially regulate reinstatement. So she just separated out her behavioral analysis of whether it was this anterior regions versus the posterior regions. And what she found is that with the drug prime-induced reinstatement, when she performed the specific manipulation of the thalamic inputs into the nucleus accumbens, she was able to recapitulate the reduction in drug prime reinstatement. So what she was seeing with the general thalamus manipulation was holding true for when she decreased those inputs. However, somewhat paradoxically, she saw the opposite effect with the cue-induced reinstatement. There was actually an enhancement in this response compared to a decrease. And when she performed this manipulation in the posterior regions, there was no effect. And so we're still, sort of trying to figure out what might be modulating that cue-induced reinstatement. But as sort of a general summary about the role of these glutamatergic afferents, if you take the cortical inputs as well as the thalamic inputs, we believe that it's this overall glutamatergic drive that's coming into the striatum that is responsible for these responses following drug exposure. And then with respect to the cues, we think that it's sort of a balance or pattern of glutamatergic activity in the striatum that may be responsible for these responses to the cues that are associated with drugs. And so to kind of give one last summary slide here, I've shared with you a lot of information that we've been finding, but what we think is happening is that repeated exposure in a subset of animals, the ones that go on to become addicts, we're seeing both a decrease in activity of the indirect pathway, the brake. We see this enhancement in activity of our direct pathway, which is our go signal. And we think that the reason these, in part, the reason that these two pathways are changing is because we're shifting the balance of the glutamatergic inputs into the striatum, which is changing activity of these cells. So I think I'll end there and just acknowledge the members in my lab, both current and past, that have contributed to this work, collaborators, especially John. A lot of these initial studies were done in his lab, and then funding sources. Happy to take any questions, if anyone has any. So we actually haven't tried ethanol studies yet. We've sort of switched in terms of looking at how similar the circuits are to look at heroin. It's a little bit easier to have the animals administer that. But our hypothesis with different drug classes is that in large part, a lot of the circuits that modulate reinstatement will be very similar with probably some nuances across drug classes. Okay, well, thank you very much. Any other questions? Not used to giving an hour-long talk. Most talks these days are like 20 minutes.
Right. Well, you have more data than you showed, which is where you want.
Right.
Is there a reason that sugar would not respond?
Yeah, so I guess one difference that I didn't really highlight is we're not doing like pathological sugar, if you will. So like there are models where you can really like sort of obesity models where you sort of cause almost compulsive sugar taking in much of the same ways that you would cause compulsive drug taking. And so kind of like how we didn't really modulate the drug responses in that normal group, I think that's why we're not totally damping down all motivation.